Okay, and we're recording. All right. Hello, everybody. This is um, CJ 352. My name is Jason Linville. This lecture is being recorded in the spring of 2020. Will probably never be seen again because 352 is typically not an online class, but anything goes in 2020. And what we're going to do today is we're going to talk about short tandem repeats, but more importantly, this lecture is going to be divided up into 3 sections. So the first section, this section is going to be on human genome. But before we begin human genome, we want to summarize as far as where we're at in the class. And where we're at in the class is, if you remember way at the beginning, we've already had an exam where I've given you permission to forget almost everything from the first half of the course. But we started with the blood stain on the board, and we were converting that into a DNA profile. So we talked about DNA, a little bit about background, the type of samples it can be found in. I mean, blood and semen are basically the 2 most prevalent in forensic science labs. And once that DNA was collected, we went through the methods for testing, presumptive testing, confirmatory testing for blood, semen, and saliva. And then we ended right before the exam with a lesson on how to extract DNA. I should have locked that door. And we basically take our evidence, our blood or our semen stains or whatever, soda can, and we collect as many cells as we can because that's where the DNA is. It's in the cells. And then we do a DNA extraction from those cells. And we wind up with what we saw at the very end of that extraction lab, where we have a small amount of DNA in a tube, and it looks like it's just water in a tube because that's what it is. All right. Our recent lecture is where we talked about how to amplify DNA, and think to yourself, what are the 4 things? See if as a class in the chat room, just as a side, if you guys can name the 4 things that go in a PCR reaction, because we're going to come back to those today. But we're not really sure as far as what area of the DNA to amplify. I think we have some clue because we know that we're going to wind up with 13 pairs of numbers. And from CJ110, we know a little bit about STRs, and we had loci and alleles, but we're going to sort of go over and reinforce that today. So we're going to talk about the areas to amplify and exactly how do we adapt that PCR reaction to amplify STR regions. Okay, so we're going to begin with the human genome. And again, I'll just mention this. I'm starting these mini lectures out, and at some point today, I'm going to stop this recording and then start another one so they're divided up a little bit when they're posted on Canvas. But we're going to start out just talking about the human genome. So in these next several slides, what I want you to focus on is, as it says there, describe the area of the genome where repeated DNA is located. So again, if I give you like a word, a microsatellite or short tandem repeat, understand as far as and when I say where it's located, we're going to see there's like coding regions and non-coding regions in the human DNA. And then also locate a short tandem repeats. So if I give you just a string of A, G, C's, and T's, I want you to be able to circle what the short tandem repeat is. Just demonstrate your knowledge as far as understanding what we're talking about. Okay, so this is a slide that we had earlier. We know that DNA is in the cells in our body. This is just a reminder that the DNA that we're talking about for these short tandem repeats, we're talking about the nuclear DNA, the DNA that's in the nucleus. Let me get my little pen here ready to go in case I need it. Yep, the nucleus. I can't even circle right. Okay. All right. So this repeated DNA, these short tandem repeats that are going to eventually lead to pairs of numbers, and we're going to look at 13 of them to give us our 13 pairs of numbers. It's found in non-coding regions of DNA. Okay, what is the non-coding regions of DNA? What exactly does that mean? Okay, what the non-coding, sorry, I'm still trying to get rid of this box. What the non-coding regions of DNA mean is that they don't code for proteins. Early on, we learned that DNA is like a recipe book for making these little nugget machines in your body. Well, STRs, they mutate a lot, and we vary in the number of repeats. If those letters actually coded for a protein, then in that area, all of us as humans would have different proteins, and a lot of us would have proteins that wouldn't function. So therefore, there's a lot of DNA in the human genome that's probably incorrectly called junk DNA. It's just we don't know exactly what it does yet. But we do know that there's free to be mutations like these small repeat mutations, and it doesn't affect us phenotypically in a way that we can see. And that non-coding region DNA is actually a large amount of the human genome, approximately 70%. So like 30% is the recipe book that is actually read. 70% of it is still there. We're still 99.98 or 99.8, whatever the number was, percent similar in those areas. But there's where we have our differences are in those non-coding regions. Okay. And of that of that non-coding region, 20% of it is repeated DNA. So it's not as though this, non-coding region is just a random mix of AGs and CTs. The reason that humans, as a further developed species, if we were to compare ourselves to bacteria, for example, bacteria, they just have genes that code for stuff, and all their DNA is very compact and it does stuff. With us, we have a lot of this non-coding regions where if mutations in large chunks of the DNA is repeated, then it just sort of builds up that amount of non-coding DNA. But again, it doesn't really affect us phenotypically. And then of that repeated DNA, or of the non-coding DNA, we also have STRs, which make up a smaller portion, approximately 3%. So in other words, there's a lot of different types of repeated DNA where you can have big chunks of DNA repeated. But STRs, we're going to focus on the repeated DNA, which is just little like AGCT, AGCT, AGCT. Okay, so the STRs that we're going to look at are in these non-coding regions. All right. So this sort of gives you an idea of the terminology that's associated. And I don't think I'm going to ask any questions about satellite DNA or VNTRs. I just want you to know down here, short tandem repeats, and we're going to go into this a little bit more. That means it can be anywhere from 2 to 6 base pairs. And we're going to find that often forensic scientists look at tetranucleotide repeats, which are 4 base pairs, meaning AGCT, AGCT, AGCT. But you could also have like a CT, 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 a 2 base pair repeat. Or you could have, does everybody know what it sounds like when you say 6 letters in the repeat? Well, let's all find out together. AGGCTT, AGGCTT. Like it could be 6 letters repeated as well. Okay. So we're going to focus on the short tandem repeats is what forensic scientists focus on. And there's a reason why these are a little bit more, are not used as much. And the reason is they're more difficult to analyze. Okay. All right, so we know short tandem repeats are in non-coding areas of DNA. That's the important part. Those percentages there, I think I do want you to know that the non-coding region there's more of it. It's the 70%. And then finally, let's look at this string of letters and try to find the repeat. Okay, this can be difficult to do because if you look at it, it's like, okay, here, AT, no, that's not it. And this GATC, oh, look at that. We hit on something right there. Okay. So eventually what you have to do is just look through this. And sometimes like G8, there can be a couple repeats and then no, it falls apart. Okay. So here we have ATCG, but also if you shift it over, TCGA is also repeated. So it's just, I don't really care where you start for this example. I just want you guys to be able to circle and recognize, hey, this is an area of DNA where there are 4 letters repeated back to back to back, and it occurs anywhere from like 6 to 15 times. Okay. All right. Really, that's a good question. Actually, let's go back because we have a good question as far as how many are repeated in a row. And what does this say? It looks like the low number is 5. And I can't see what the high number is because I have a little Zoom screen that has frozen in part of my screen, but it is 5. So I guess if there's 4, that's no good. Okay, and if there's 41, 5 to 40, thank you, Elise, then that's too much. Okay, but it's typically we're going to see the numbers that we get tend to start around 6, really 7, more prevalent, and they can go up normally to like about 16. But some STRs that we look at, the range is more like from 20 to 40. So it's a little bit higher than that. Okay. All right. Let me get back to where I'm going. Okay. And then finally, fun facts. These, I'm not going to ask you any questions about this, but it's just a reminder, 3% of the genome. And this gives you an idea of, how often they occur. I don't really care about that. The one number, and the reason why I left this slide up here, is to recognize that there were about 8,000 STR loci. So when forensic scientists look at 13 loci, recognize that there were like 8,000 that forensic scientists could pick from. And the FBI was actually the group that organized and said to every state in the United States in the mid-90s, when STR analysis, it was obvious this was going to become a major part of crime labs. The FBI said, okay, hold on. They work with commercial companies, and they said, these are the 13, because they don't want Alabama to analyze 10 loci and California to analyze a different 10, or for one company to sell a kit to do this and another company to sell a kit to do a different set. They wanted all state labs to be compatible. So the FBI actually selected 13 out of those 8,000 for analysis. And STRs were not once married to Elizabeth Taylor. This slide was had a little bit too much white space down here, so I wanted to fill it in with a fun fact. And whenever I think about fun facts, I think about Elizabeth Taylor. That's not true. Okay, that's it for the human genome. So what I'm going to try to do here, despite this challenge of getting through here, I'm going to stop the recording for the human genome. Thank you very much for listening. Have a good day.